Cellular respiration can be divided into two kinds, aerobic and anaerobic, in other words, with oxygen and without. In this series, we're going to be looking at aerobic respiration, and we're going to look at the steps needed in order to access the energy that is stored within sugar. Now, there are three key stages, which is glycolysis, the Krebs cycle, and oxidative phosphorylation. And I'm going to be making a separate video for each one of these that you will need to watch in succession, just so that you have a very clear understanding of what's going on in each step. Because you cannot jump a stage, you have to watch it in the correct order in order to know where the lesson goes from here. So to make it really easy to understand, we are going to break down glycolysis, Krebs, and oxidative into their main functions and their purpose. And starting off with glycolysis. Glycolysis is the step where we are taking a sugar, hence the name glyco, and we are cutting it, lysis. And we are preparing the sugar in order to access the hydrogens that are attached to the sugar. And we're going to look at the structure of sugar soon so that you can see what I mean by that. Once we have the products from glycolysis, we can go into the Krebs cycle. Now, the Krebs cycle is responsible for harvesting those hydrogens. So we make them accessible in glycolysis because we want to access them. In the Krebs cycle, we harvest those hydrogens, and I'm going to explain as well why we need to do that. And once we've harvested all of these hydrogen atoms, we use it in the final stage called oxidative phosphorylation. Here, we use the hydrogens to make the energy carriers ATP, which, remember, is the goal of cellular respiration, to take sugar and oxygen and to create carbon dioxide and ATP along with water. So that we have a very good understanding of how glycolysis works, we need to know what the shape of the glucose molecule looks like. And what's most important is the hydrogens that are sitting on the outside of our molecule. We want to access those. Why? Because hydrogen is an atom or an element that is very energized. It bonds very easily with other elements on the periodic table. And because of the bond it does eventually form, it sticks other elements together really, really well. It makes a very stable molecule. And what we want to do is we want to access these. Now, right now, this glucose molecule is very stable. So it's really hard to get at these hydrogens. They're not just going to fall off. And that is where glycolysis comes in. We are going to cut the sugar up into pieces so that when we're done, we are going to be able to access the hydrogens a lot easier, because right now in this big structure, it's really hard to get at the hydrogens. Glycolysis begins with a glucose molecule that has 6 carbons, and each of these pink circles represents one carbon atom that is attached to another. Right now, this sugar is far too stable for us to access any of the hydrogens that we want. So what we need to do is we need to make this sugar molecule unstable. And imagine it being like a seesaw. If you've ever played on a seesaw before, 2 people sit on either ends of the seesaw and they make it unevenly weighted so that the seesaw goes up and down on either side. Similarly to our glucose molecule, we want to make our glucose molecule seesaw backwards and forwards so that eventually it actually breaks in the middle so that we can get to the hydrogens that are locked away. Now, the only way to do this is to add something to it so that it breaks down into a component that we can use. So in order to take us to the next step, we need to add something. And that's something that's going to swoop in and deliver is going to be our ATP molecules. Now, before you ask me, well, where did the ATP come from? I want you to think of this like a baking show. We made them earlier, and I'm going to show you where they eventually do come from. But for now, let's just pretend that these ATP molecules exist. So what exactly are they delivering? They are delivering their phosphates, and they're going to stick their phosphates onto the end of this glucose molecule. And this is what it will look like. The phosphorus is represented by these green atoms that I've added to the ends of our glucose. Now, when we add the phosphorus to our glucose molecule, we end up making a substance called ADP. And that is when we lose a phosphate. Now, how did it change its name? Well, ATP stands for adenosine triphosphate, meaning there are 3 phosphates in the beginning. But once we lose a phosphate, we are going to end up with 2 phosphates, which means we now call the molecule ADP, or adenosine diphosphate. This little ADP goes off back into the cell, and it's going to collect another phosphate so that it can turn back into ATP. But now that we've made our molecule unstable, we have a process that has occurred, and it has a name. This whole process of adding a phosphorus to our glucose molecule is known as phosphorylation. And it gives it away because it literally has the word phosphate in it. Now that we have created a molecule, it has a new name because we've changed its structure. And this new molecule is called glucose diphosphate, which is very obvious why it's changed its name, because we've added 2 phosphates into it. Now that we have added these phosphates, it makes the glucose molecule very unstable. And so what ends up happening is our glucose diphosphate breaks down into 2 slightly more stable molecules, and they look like this. These are called pyruvic acids, and as you can see, we've formed 2 of them. There are 3 pink carbons and 1 phosphate at the end. And so it's important to know that these are 3 carbons in each, and as you can see, we've just halved the glucose molecule into 2. Now, at this point, we have come to the end of glycolysis. And the whole process of glycolysis takes place in no oxygen. Now, this is something that everybody gets wrong in exams. They forget that glycolysis has no oxygen present when this happens. As you can see, there's no oxygen moving through this process whatsoever. And so what we are doing here in glycolysis is we're taking a glucose molecule, we are investing some energy into it to break it down. And so in this beginning stage over here, where we added the ATP, we invested a little bit of energy. That energy is coming from the phosphate that binds to the glucose. That energy makes it so unstable that the glucose diphosphate breaks down into 2 pyruvic acids. Now, up until this point, no oxygen has been used. If there is oxygen present, we will go into the next step. Please note, what I'm about to explain now is not directly part of glycolysis. It is only going to happen if there is oxygen present. It's like a little in-between step that occurs in order for glycolysis to move into the next phase. Now, this is an important key note that you need to make. What I'm about to explain to you is an in-between stage where we move from glycolysis to the Krebs cycle. This little in-between stage only occurs if oxygen is present. So, if oxygen is present, then the following things are going to happen. We're going to take our pyruvic acid, which we can see right now. We've got our 3 carbons and our 1 phosphate, and we are going to lose some atoms. Now, that's okay, because what we're trying to do is we're going to try and make a compound that we can actually use to harvest even more hydrogens. And so what we're going to do is we're going to take our pyruvic acid, and it's going to break down, and it's going to lose 3 things. The first thing that is lost is going to be hydrogen, which is great because hydrogen has a lot of energy, and we're going to use this hydrogen at a later stage. Nothing is wasted. The next thing that is going to be created, actually not lost in this instance, is going to be ATP. And ATP is created because, if you remember, this little green round circular ball at the end here is a phosphate. So we use that phosphate and we stick it to an ADP. So essentially what we do is we go ADP plus that little phosphate gives us the ATP at the end. Now, the third and final thing that breaks off at this point is some carbons, and those carbons attach to the oxygen, which must be present, and it forms carbon dioxide. Now, this carbon dioxide is going to be exhaled. We have no use for it, but the hydrogen and the ATP have an important use. We are going to use those at a later stage. Now, what is the substance that we are left with? Because now we've broken down, we've pulled off phosphates, we've pulled off hydrogens. We are left with a substance called acetyl, which has 2 carbons left. And it looks something like this. We've got 2 carbons left over. We've lost a carbon to carbon dioxide. We've lost a phosphate to ATP. And we have collected some hydrogens. Now, acetyl needs to be moved around. It is a molecule that we are going to use to harvest hydrogen off of, and we need to move it around. Now, as we know in basic biochemistry, all chemical reactions are sped up by enzymes. And there are also things called coenzymes, which are very, very similar to enzymes, and they assist with chemical reactions. In particular, acetyl combines itself with a very particular enzyme, or should I say coenzyme, called coenzyme A. And when you put the two together, they form a substance called acetyl coenzyme A. You basically just put the whole name together. This product that we get at the end, acetyl coenzyme A, is the first substance that we use to enter the Krebs cycle, which, as I said, will be in a follow-up video from this one now. So remember, what was the purpose of this whole glycolysis step? To access the hydrogen. We wanted to take a very stable sugar molecule, break off the hydrogens, break off the carbon dioxides, get some phosphates so we can use it for ATP production, because at the very end, what we want to do is we want to pull off as many of those hydrogens off the sugar as possible. If you need to remind yourself, go back to the beginning of the video where I spoke about the structure of the sugar, and you'll see all the hydrogens on the edges, and we want to pull those off, and we do all of that in the next step, which is the Krebs cycle. So as always, let's summarize this lesson into the terminology that we have learned. First of all, we looked at the 3 steps of cellular respiration. We looked at glycolysis, which is the step where we are preparing the sugar. The Krebs cycle, which we will do in the next video, which is where we are going to harvest the hydrogens attached to the sugar. And lastly, oxidative phosphorylation is the process where we take the hydrogens we have harvested, we use their energy to create ATP molecules. Now, to do glycolysis, we start off with a glucose molecule, a sugar molecule, and we make it unstable by adding phosphates through a process called phosphorylation. We add some phosphorus to our glucose, and we create glucose diphosphate. So there are 2 phosphates that are added. Where did those phosphates come from? They came from ATP molecules we've made earlier. Don't worry about the fact that they were made earlier. Just know that they are there for now. That glucose diphosphate is a very unstable molecule, and so it breaks down into 2 pyruvic acids. Those 2 pyruvic acids are 3 carbons each with 1 phosphate. Now, it is at this point that glycolysis ends, and I want to reiterate that once more. Only if oxygen is present can we go into this little in-between step to prepare the pyruvic acid for the Krebs cycle. Now, this in-between phase is when we take our pyruvic acids and we prepare them just a little bit more. The first thing is we're going to take some hydrogen off of our pyruvic acid, which is a good thing because we're going to use that hydrogen later on with its energy. We are going to create ATP, and we do that by taking the phosphate that was originally attached earlier on, and we create ATP, and we are left with a substance called acetyl. It is a 2-carbon compound. Where did the other carbon go? It was lost via carbon dioxide. So we went from 6 to 3 to 2. Now, acetyl cannot move around on its own, and we need to speed up this reaction so it's not too slow. So we introduce coenzyme A. Think of it like a wheelbarrow. It picks up the acetyl and it carries it to its next destination. And that is where the Krebs cycle will begin. As always, everybody, I hope that you have loved this video. Make sure to give it a thumbs up and subscribe. Turn your notifications on. I post a new video every Thursday covering different topics for high school level biology, and I'll see you again soon. Bye.